Okay, so we just finished this, yeah? And now we're going to start moving into another type of junction. In this case, we're going to move to the apical region, that is the tight junctions. That, sorry, my brain is tired. This is the one that they have the blood brain barrier, that is very important. That also, for example, in your stomach, what happens if all the enzymes of your stomach go to the lungs? They will destroy the lungs. Yeah, so you will need to have very strong tight junctions that don't allow that the enzymes from your stomach go to other places. So they have to have very good tight junctions. Okay, and I will quickly talk about gap junctions. They will be you already know this the information. I'm just going to tell you the structure. In the last class, after reading week, I will talk about the extracellular matrix, and then I will go again a little bit about the cell wall, yeah, and compare them. In your textbook, they show that there are some similarities. Okay, so let's start. So um, the tight junctions, I will tell you that they work as a barrier and control of paracellular permeability. So here, this is why it's so important. It's a barrier, and you can find it in the kidneys, in the tight junctions, and they uh, control the paracellular uh, transport. Yeah, so the transcellular is the one that requires endocytosis that go pass through the cells. The paracellular is the one that cross between the cells. You remember what the white cells did, opening the space? That is not common. Yeah, that is not common. But so you have tight junctions there that avoid that, and in that way, bacteria doesn't go into our brain. Enzymes of our stomach don't go into the lungs or into other tissues. Yeah, because we have these tight junctions that hold the cells tightly together, let's say. And I will talk about the components. So they will be, again, core, then a linker proteins, and the cytoskeleton. Same thing. And so you will learn about three main proteins: claudin, JAMS, and occludin, and adapter proteins that they will be the sona occludins. Okay, so uh, pretty easy, Julia. Could you help me with this one? Hopefully, I didn't make a mistake. Yeah, so the tight junctions are cell-to-cell adhesion structures that create a field and a filter between adjacent epithelial cells. Okay, so here, good job, Julia. So here they show a diagram in your textbook that I really like it because they combine um, different views of the cell. So here you can see your cell from the top. Imagine that you go to Google Map and you go on a city and you zoom in and you see all the ceilings of the houses. Here you're seeing the ceiling of the cell, and the cells have polygonal shapes. All the epithelial cells, yeah, they look beautiful, and for some reason, uh, just physics, they have similar to bubbles. When you put soap bubbles, they most of the bubbles will have hexagons. Here, honeycomb patterns are the more common pattern that you can find in the ceilings in this area of the cells. Here you can see it from this side, from this lateral side, and you can see that it's a belt, but it's not covering the entire region, only the apical region. So you will see one here, another one here, and another one here of this lateral region. So we went from the top, from the lateral region, and remember this is a belt, and here they combine it with electron microscopy pictures, and here you can see, do you see all these are loops in the cells. If you remember the NCAM cells, that they have loops. This one way also will have loops, and they will be very important for the filtration of some ions. Okay, so they generally don't allow anything to go through except for some structures that form loops that I will show you in a sec. So what are tight junctions? Are intercellular adhesion complex that they are found in epithelia, and endothelium. And remember, this is the red structure. Yeah, they are like a belt. They are so they surround the entire cell, so they cannot go through any of the spaces. Here they only look like a dot because we are only seeing this part. We can, if we put all the images together, they will form a belt. Okay, so Alicia. So I don't know if what happened if I do this experiment and I put with a very tiny pipette, I put a dye here, and there is a tight junction. Is the dye going to go through the cell or is it going to stop? You know, something that color, and I put it through the cells in the space in the intercellular space. So that is very important. So it's going to go start what they did that experiment, and what they found is that when you find a tight junction, yeah, the tight junction is going to stop any of those molecules, yeah. So here you have the tight junction, the basolateral surface, and here they have a solution or a tracer molecule, and here they are showing that it's not able to go through that. Here, this is just the electron microscopy picture, and I just made it easier for you to see it. So they are very good at blocking things. Yeah, so epithelial cells need to be tied together, and the tight junctions not leave any space, and they are very important. So now we're going to start zooming in into a tight junction. Okay, so perfect. Julia, this is what you said. And Zia. Okay, so here you have a cell with tight junctions. There is a clouding and uploading. Yeah, these are two proteins of the tight junctions. And here you see a cell that don't have those proteins. So tell me what is the difference that you see in this diagram between a cell that have tight junctions, two cells that have tight junctions, and two cells that don't have tight junctions. The one that doesn't have the tight junctions, letting the macromolecules yeah, so this one, all the macromolecules are going. So this could be sugar, DNA, it could be a virus can go there and infect all the body. Here they have water and sodium, so ions and water, and so they selectively can go through some of those. Yeah, so they don't allow that these big molecules, macromolecules, go through that. These two proteins are blocking everything. So perfect. And remember, this is paracellular. Yeah, when it's in between two cells, it's paracellular. When it's in between, when it passes through, is transcellular. Okay. So function control the paracellular permeability, and then it's forming a continuous belt. Yeah, and so the molecules will only be able to pass through the cells. So they need to have here endocytosis, and they will be able to only go through there. Yeah. So in that way, you control, and uh, this is another proteins that are important in uh, the immune response. So a bacteria is not going to get in. Yeah, and the tight junctions are protecting against that. Here you can see a diagram. You know, this is beautiful. In my computer, this is green and that is brown. Um, yeah, and this color change a lot. I cannot believe how weird are computers. But anyway, so here, if you see in red, you have a lot of proteins, and those proteins are involved in trying that the glucose go inside of the cell. Here you have the tight junctions, yeah? So the concentration of glucose flow here, but the glucose can get into any of those proteins in these transporters. And here you can have higher concentration of glucose, but there is a transporter here there is no transporter here in the tight junctions. There is an area that is blocked. There is nothing that goes through there. Yeah. So the tight junctions confine the transport of proteins. Yeah. And they act as a diffusion barriers, and they prevent the black flow. Okay, the backflow. So here you can see the diagram that I was telling you. Two type of transport: paracellular, in between cell, and transcellular that pass through the cells. Okay, so here you find the definitions. The tight junctions, TJ, are very important for the transcellular junctions. This is a diagram taken from your textbook. Here you can see how some. Okay, Maddie, why there is a line, blue line there? What is indicating? And that way, but we said that they are blocking a lot of things. So why there is something going through? Perfect, perfect. I love your multitasking. So here, but there is no backflow that it can go in the other direction. Yeah. So is in the same way that there is a selective permeability in the lipid bilayers, here you have also a selectivity here, and only ions and water, and I will show you how this is done in a second. So here you have the intercellular space, the two plasma membranes, and here you find the ridges of the tight junctions. You can find all this diagram with all the names also in your textbook in case that you wanted to see it. I already explained what is the paracellular transport that is go in between this intercellular space, and that the paracellular barrier that the junction can block some molecules to go through there. So one good example is that the tight junctions are blocking the lateral movement of the of membrane proteins. So again, imagine here you have a lipid bilayer, you have proteins. So here what is happening is that here you will not have more proteins. These proteins are not going to be there because that area will be blocked. Yeah, so you have two type of membranes. The outer membrane here, yeah, this outer membrane, and there the lipids and the proteins, they cannot move laterally, they are blocked. But this, and in that way it works as a barrier, and in the inner only the proteins. So this area will be blocked. So in that way, there is nothing happening here. Okay, so now I just told you what it was a tight junction. I told you that they were multi-complex of proteins, that they have a function that is hold the cells together, and that they prevent or the trans or control the paracellular transport. Yeah, and Zia explained and Maddie showed us, okay, there are some molecules that they can go, but other ones they cannot go, and they prevent the black flow. They block the backflow. Here you can see the different components. So again, uh, Eric, this I always repeat the same thing. Could you see the three components of this junction? Jams, clotting, and occluding. Remember what are these ones? These are the core. And this one, this zonula 1, 2, and 3, this one will be linkers or adapters. And here our friend actin will be, in this case, is the cytoskeleton, and it will be instead of intermediate filament, this will be actin. Okay, so very good. The desmosomes have intermediate filaments, but the adherent junctions and the tight junctions have actin. Okay, yeah. So again, the same thing. Of course, they will change the names, but it's the same idea. So, um, mhm. So here um, different from other ones. The idea here is that they make a strong and tight seal. So they have this network, yeah, of proteins that are blocking the 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 path of molecules. So this is why you have the clotins and the occludins with those structures, and each one of those strands is embedded in the plasma membranes, and they have extracellular domains in those strands, and they also have, again, as I just said to you, the zonula adherents that are the adapters or linker proteins, and then those zonula adherents will link to the actin. And there are three major proteins that I just showed you. Clotting, these JAMs, that means junctional adhesion molecule, and finally the occludin. And I just told you that the zonula occludins, these ones that are three, 1, 2, and 3, are the ones that connect the core proteins to the actin filaments. So, Eric, just in this diagram, I just wanted to show you exactly what we just talked. So, again, core, uh, here you have the trio of, of proteins here, and here actin is there. And again, core, linker proteins, and here, as you said, the intermediate filaments are here. Okay, with keratin, ventamin, all those intermediate filaments. Okay, so those two are similar. Adherent junction and tight junction, and similar. They have actin. Desmosomes will have the intermediate filaments. Okay. I don't know where were we. Oh, Duncan. Okay, super easy. Which proteins are involved in the formation of tight junctions? Yes, that even I that I'm really tired right now. I perfect. So we have clotins and occludins, okay? And the coherents are located, could be the E coherents will be in the adherent junctions. You can also have desmosomal coherents that they will be in the desmosomes, and the integrins will be in the basal membrane. The okay, somebody knows this collagen and elastin where are located? Collagen and elastin. Do you know that they always find it in some creams that they, if you have collagen, your skin will look good and you will look young forever? Collagen and elastin, we will talk about that. They will be in the extracellular membrane. Okay, so in after reading week, we will talk about these ones. Uh, so here, so I hope that this is now very clear. Yeah. Now we're going to zoom even more. Yeah. Now we're going to zoom in the clotins. So you remember that they have these uh, domains, extracellular and transmembrane domain, that they go back and forth, all these strands that they interlock. So here, if we zoom in, you can see one here, or here, or here, yeah? And you have an extracellular domain here that they form two loops. Those loops are important. Those one are the one that create the filter. So the one that Maddie and Zia told that ions are blocked is because of this extracellular domain. That is the function. So let me show you. That is in your textbook. So here you see again another diagram that makes the life easier. Here you see in the transmembrane domain. Here you see extracellular loops in the clotins, and here you see that those one will touch. Yeah, and the main function of those one structures is that they are ion selecting ports that allow the passage of specific ions. Okay, so in addition to have these very tight junctions, these loops, the big loops, are the ones that produce this selectivity. Okay, so just to finish, I talk about tight junctions that are the ones that are in the apical region in the top cell. Then I told you that the tight junctions, they held the cells together, and they form a barrier that controls the paracellular permeability. I also told you that this is what they are controlling. This has to go through endocytosis. I also told you that there are different components, and I told you that there are three core proteins: claudin, JAM, that is the junctional adhesion molecule, and the occludin. And I told you that the claudins are the ones that are interacting, producing those loops that produce the ions of selectivity, and the adapter proteins or ligand proteins will be the sunlight occludins and the ACT2 actin filaments. Okay. Uh, oh, Angus, I think easy question for you. Gap junction. Okay, so A, what is A?
That one is
Very good. B, definitely not D, and definitely a structure that anchors cells to extracellular matrix. Which one will be that one? That will be an integrin, yeah, or a Henle desmosome, or yeah, focal adhesion, yeah. So this one, and Angus, perfect. So that one will be correct. Yes. So now to finish the class, quickly I'm going to talk about the other junction here, that is the gap junction. And I will be very happy if you remember that the gap junction are small pipelines, very tiny ones between cells. So the plasmodesmata, big, big ports, and I told you that in fungi, you know, this the communication, you can bring even nucleus and also the mitochondria and organelles. This one, only ions can go through that. Yeah. So there is communication, but only ions can go through that. And this one are the only one that have no cytoskeleton connection because it's only for communication. And the function is related to uh, the communication of ions during for the electrical response. And I will tell you the structure. So different from the other protein, you will have just this simple intracellular channel that is formed by a tiny molecule that is called connexin. That when you have an hexagon, you call it connexon. Yeah, when you have six of this. So this is a hemi-channel because it's only half. When you have 6 and 6, you will have the entire channel. So this will be for one cell. For the two cells to communicate, they will have 6 and 6. They will have 12. Okay, and you can have different combinations again to be homeotypic or to be heterotypic. Okay, so let's quickly go into that. Again, one cytosol here, another one cytosol here, and again, here is only ions, the one that are going to cross. No nucleus, no organelles, no DNA. Those ones are very tiny. Yeah, so gap junctions are regions where the plasma membranes of cells are aligned. A gap junction has very small gap. They are small pipelines, and the function is this electrical and chemical communication. Uh, and this allows that small molecules and ions pass directly from one cell to another. Uh, Again, this is a diagram taken from your textbook. Cell 1, cell 2, and here you can see the channels, all the channels together, yeah, that cross from one cell to another one. And I told you that these big channels are called connexons, the one that are the full channels, and this one full channels have 6 subunits of connexin. I always, in this introductory biology courses, we don't look at the plants almost. I'm talking about only about the cell wall and the plasmodesmata. We mainly focus on vertebrates and humans, mainly that are the most of the applications. In invertebrates, they don't have many of these proteins. They have a similar one. So in the test, this one can appear. This is called, they will have annexin. And in invertebrates, they don't have tight junctions. They have subtight junctions, but they have the similar function. So again, so check this one. Invertebrates have annexin instead of connexin. Here you can see the schematic. Here you can see the diagram. They are so tiny that this is the electron microscopy, and you can just see the proteins there. And again, connexin there. One connexon will have 6, and they don't allow the pass of proteins, nucleic acid, or organelles through. So this is different to from the plasmodesmata. Okay, so with this we finish. I just finished the gap junctions. Remember, small pipelines, they only allow ions to go through that, not big organelles, not the nucleus. And the structure, they're called, the subunits are connexins. 6 connexins will form a connexon, and they form a hemichannel. When you have 2 connexons, you will have the intercellular channel, and here you can have a lot of variations in how they mix, how they form. Okay.